Very good afternoon, children. Very good afternoon to all. How many of you are there, children? Can I just send a message who are all there? Who are all there? Can you send a message? Laya is there, Setu is there, Sachin is there. Laya, Sachin, Setu. What about others? Okay, children. Very good. So let's start the topic. Before that, I want to discuss something with you regarding your preparation for the NEET. Regarding the preparation for the NEET. Can you tell how many days are there for you? Can you tell how many days are there for you, children, for the NEET exam? Nearly 60 days. Okay, nearly 60 days. What about others? "58 days. Okay. Okay, children. So I'm going to give a plan for you how to go about. How to go about. I'm going to give a plan for the rest 59 days. The rest 59 days. The plan that I want you to execute is the next 59 days. It is very crucial for you. Very crucial for you. So all of you listen. Take a piece of paper with you and make a note what I'm explaining here. The next 30 days, I want you to concentrate more on class 11 portions, class 11 syllabus. For the next 30 days, I want you to concentrate more on class 11 syllabus. Class 11 syllabus. Because the class 11 syllabus, excluding the first chapter, the second chapter, morphology, it is a very big lesson. And then cell structure is also a very big lesson. And after that, plant physiology. Plant physiology and animal physiology. these 4 chapters in class 11, it is more important. More important. I'm not telling the diversity living organism is not important, because you'll get more questions from these chapters. You'll be getting more questions from these chapters. More questions will get from these chapters. That's the reason why I'm telling this chapter is very crucial. Very hardly in diversity in living world will get 2 or 3 questions. But out of 45 questions from 11th standard syllabus, you are going to get from this. And these chapters, it has more weightage when compared to 11th standard, when compared to the 12th standard, when compared to other chapters. Morphology, cell. How to prepare these chapters? How we have to take yourself in this chapter? You have internal choices are there. That's what they told. But if you are thorough with the concept, then only you can take either or. So these 59 days, 30 days allot exclusively for 11th standard syllabus. Morphology, cell, plant physiology and animal physiology. And what you are supposed to do it is here in all the chapters, NCERT syllabus, NCERT textbook. Line by line, you have to be thorough. Line by line, you have to study and understand what is given there. Line by line. Understand all the concepts that is there. That is the foremost thing what you are supposed to do it. If suppose you are studying a chapter cell biology or cell cycle or cell division, cell cycle and cell division. Take a complete day, or if you're going to plan it for 2 days, what you're supposed to do is the 2 days learn theories first, and then attend MCQs. For each chapter, minimum of 10 questions, 100 questions, you have to guess exposed. Without reading, don't write the test. Don't write the test. Because if you are attending around 100 questions, out of these 100 questions, at least 80 plus question you should know answer. Then only you'll be more confident. Suppose you are not learning it properly, if you are going to attend the test, out of 100 you will get 35 plus questions correct. Then definitely it will put you down. Your confidence level, it will come down. Plan according to that. 11th standard, since it contains more portions there, I want you to allot 30 days for that, exclusively for that. The next is the next 29 days. The next 29 days. Already you're all well versed with 11th, 12th standard syllabus. In that 29 days, allot 15 days for class 12 revision. Again, the revision, you have to go NCERT line by line, right from intro to summary. There will be a lot of examples in 11th standard. Make a note of it. When you are writing, when you are learning some study materials, will come across the year in which it has been studied. The scientist who discovered that, what he has discovered. Like that, I want you to take notes. 15 days, complete revision for 12 standard syllabus. Line by line, again, we have to take MCQs. In the MCQs, for example, if you put the topic digestion and absorption and you search for MCQs for NEET, there are n number of questions it will be there. Attend those questions. Even if you know the answer, just go ahead. If it is wrong, find out why it is wrong. Then you will come across the correct answer. Then you will understand what is the correct answer there. And the rest in between that. So now almost 45 days over. The rest 13 days odd 13 days or 14 days are there. These 14 days take full paper test. Full paper test. In the net, the internet, there will be some mock test conducted by NTA or some other websites. They will be conducting certain mock tests. Attend those mock tests. Try to answer there. Get exposed to that. Children, in each chapter, if you are going to attend 100 questions, in each unit, if you are going to take for about 200 to 300 questions, definitely children, it will be very good for you. And also revise the previous year question banks. There are previous year question banks are also available. Please go through it. Previous year question banks are there, right? Those papers are very important. Please go through that. And moreover, be confident. Be confident in yourself. Never lose hope. But without doing smart work or without preparation, it is not possible. There is a scripture word which I like it very much. Oneness. The horse will prepare for the battle, but the victory comes from the Lord. The horse will prepare for the battle, the victory comes from the Lord. That means you have to prepare yourself. Have faith in God. Have faith in God. Have faith in God. Put to a fullest effort. Fullest effort. Then definitely you will succeed. Don't worry about if you are losing marks somewhere. Everybody will learn through mistakes. If there is a mistake, rectify that. If suppose you feel that, sir, I am not confident with this particular topic, only you know better about yourself. Only you know better about yourself. Liar knows your strength. Liar knows where you are lagging. So you must understand your negativity and how to change that to positivity. All those things are very important. The nearly just 59 days or 60 days or 58 days, whatever days are there, take very good sleep, nice sleep, study well. Whenever you feel like sleeping, sleep, children. Sleep well. But see to that, you are compensating those time in reading. At least we require minimum of 13 to 15 hours of learning on these days. Not only biology I'm asking, I'm telling you to follow that in physics and chemistry also. But biology you can easily score 330 plus. Exclusively in biology it is possible. I'm not telling you it's not possible. Other topics, others is also possible. But here, most of the questions are, if you are thorough with the concepts, you can immediately answer to that. The questions it will be like that. So that's why. Yes, children. Okay. So this is what I want you to understand. Now we'll move on to the questions. We'll move on to the questions. One minute. The file is not opening. Just I'll check. Let me check. Okay, the first question for the day. First question is here. So you can answer to the question. All of you must answer to the question immediately. Read the question, take few minutes, few seconds and answer to the question. The factors which help in development of transgenic animals are: the factors which help in developing of transgenic animals. First of all, we should know what are transgenic animals. We should know what are transgenic animals. The animals where the genes are altered. Genes are altered. Organism in which the genes are altered are known as transgenic animals. Are known as transgenic animals. Okay, they are known as transgenic animals. In transgenic animals, there are 4 options given here. New gene transfer techniques. New gene transfer technique. Yes, children. New gene transfer technique. Here I told genes are altered, genes are transferred. So that is there. Introduction of cloned gene into the fertilized eggs. Here When the genes are altered, when the genes are inserted, they use vectors. Vectors. And the genes that are inserted into the vector, we call them as cloned genes or recombinant genes. This is also there. Successful implantation of modified X into the receptive female. Because here in transgenic animals, the whole process, it occurs in vitro in the laboratory, and then it will be transferred into the female body, reproductive tract. So all the three are there. So answer for this is all of these. All of these. The next question, RNA interference. RNA interference. It is essential for RNA interference. It is essential for cell proliferation, cell defense, cell differentiation, micropropagation. The four options are there. The RNA interference. First, you should know what is RNA interference. The RNA interference, it is a mechanism in mammals, mainly to protect the cells against certain pathogens like virus. Protect the cells against the viral RNA. So this mechanism, when there is any viral RNA or viral infection is there, the virus, it injects RNA into our body. So this mechanism, RNA interference, it interferes with this RNA and it attacks them. It attacks them. So RNA interference, it is a very efficient tool to send out the specific genes which are from the viral infections. So basically, the RNA interference, it deals with cell defense, protecting the cells from viral infections. The next question, which of the following plant pests is now controlled by RNA interference technique? RNA interference, I have told you that it is to protect the organisms from infections. The RNA interference, it is mainly to prevent the infection caused by nematodes. They'll use against nematodes. Here, the RNA interference, it is mainly, it causes impact in the nematodes, because this was first studied in nematodes in the year 1998. In the 1998, Mello, a scientist who studied about the roots of tobacco plant. The roots of tobacco plants were infected by a nematode. To protect that, they use the RNA interference technique. So the nematode that is used are this one, the Melidosine incognita. This is the nematode that is used in RNA interference technique. The next question, one of the following can be produced by genetically modified E. coli. A genetically altered E. coli, one can be produced from here. Molting hormone. The molting hormone, it is called as ecdysis. Ecdysis. This is a natural hormone. Natural hormone. The next is FSH, the follicular stimulating hormone. This is produced from the adenohypophysis of pituitary. Adenohypophysis of pituitary. Mainly the FSH, it controls the production of ovum. Whereas the E. coli bacterium, Escherichia coli bacterium, it is genetically modified to produce interferons. What are interferons? Antiviral proteins. Antiviral proteins. They are used to produce antiviral proteins. For antibiotics, for the production of antibiotics, there are various organisms are there. We have penicillin notatum, streptococcus bacteriums are used. So E. coli are mainly used for production of interferons, the antiviral proteins. Next question, DNA fragments are, there are 4 statements are given here. Negatively charged, neutral, either positively or negatively charged, depend on their size, positively charged. This is a direct question. A very direct question. Let us see who are all answering to that. (5A). Because the direct question, we all know that DNA, it is negatively charged. Negatively charged. The next question, a foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using. They're telling an information that both the foreign DNA and the plasmid, the vector, they were used to cut by the same restriction endonuclease enzyme. It can be joined to form recombinant plasmid. They are joined to form recombinant plasmid. So here, think that this is a plasmid, and then to which we are adding the desired gene. Now this, it will become a recombinant plasmid. Here the question is, which it is used to join? Which one is used to join here? EcoRI1. EcoRI1, this is an endonuclease enzyme. It used to cut the segments of DNA. The next one is TAQ polymerase. TAQ polymerase, it is a thermostable DNA polymerase enzyme which are mainly used in PCR technique, polymerase chain reaction technique. Polymerase 3, an enzyme that catalyzes the elongation of DNA chains. DNA polymerase 3, it is used in elongation of DNA. But the DNA ligase, this is called ligase. This ligase is called as paste enzyme. It's called as paste enzyme. So the paste enzyme DNA ligase is used. This is the answer for question number 6. Next question. Some of the characteristics of Bt cotton are long fiber and resistant to aphids, medium yield, long fiber and resistant to Pt parts, high yield and production of toxic producing crystals which kill diphtheria pest, Bacillus. For that, for this one, for the production of protein crystals, they are using the organisms which are taken from the genes, the Bt gene that are taken from Bacillus thuringiensis. Whereas this particular question, the characters of Bt cotton is mostly the Bt cottons are attacked by the bollworms. In the fruit, you can see there will be small holes, it will be there, which are caused by the bollworms. Holes. It will affect the whole cotton. So the answer for this, high yield and resist to bollworms. High yield and resist to bollworms. The next question, question number 8. Question number 8. Laya, want me to explain question number 4 again? Okay. Which of the following can be produced by genetically modified E. coli? The molting hormones are not produced. Follicular stimulating hormone are not produced by E. coli bacterium, whereas the interferons, the antiviral proteins are produced. Okay, genetically engineered antiviral proteins are formed mainly by E. coli bacterium. Antibiotics, they can be produced not only by E. coli bacterium, Streptococcus, Staphylococcus, certain fungus. All those things are used in production of antibiotics. Okay, we'll move on to the next question. The genetic defect, ADA deficiency, may be cured permanently by the ADA deficiency. The ADA deficiency, that is called adenosine deaminase enzyme. There is a gene that is responsible for the formation of ADA enzyme. If there is a disorder in the ADA enzyme, it can lead to deficiency of ADMA. What are the methods are there to treat them permanently? Usually it is done in babies. Usually it is done in babies. Usually it will be done in babies. How it is done, I'll just show you. In the case of ADA deficiency, the main thing is they'll go with transplantation of bone marrow. transplantation of bone marrow, where you can see it here, the bone marrow from the child is removed and the bone marrow is enriched with the parent cell of the immune system, and the defect, it is identified and the cells are infected with vector containing corrective genes. Here, why you are taking the bone marrows? Because the bone marrows are defective, abnormal is there. So we are taking that bone marrow, and that bone marrow is altered with the vectors containing corrective genes. So now you can see it here that bone marrow take up the gene, and then this, it will be the cells, the bone marrow cells, after altering the gene, it will be inserted into the baby. It will be inserted into the baby. Now what will happen? The child, after 2 weeks, they start developing ADA. So which is the best treatment that can be given here? The best treatment is introducing isolated genes from bone marrow producing ADA into the cells at embryonic stages. This is the correct answer. Of course, bone marrow transplantation is also there, and enzyme replacement therapy is also there. Administering adenosine deaminase is also there. But the question here is permanently cured. How it is permanently cured? Permanently cured is mainly altering the bone marrows, because the defect is there in the bone marrow. That bone marrow need to be changed. That is the answer for this particular question, question number 8. We'll move on to question number 9. Which of the following statement is not correct? Which of the following statements are not correct? The first one, a nematode Melodicte infects the roots of tobacco plant, causes a great reduction in the yield. Children, here I have told it already about RNA interference. RNA interference. So this is the correct answer for this is the correct statement. But here you have to find out which is not correct statement. RNAi takes place in eukaryotic as well as prokaryotic cells. Usually the RNA, it occurs in prokaryotic cells that deals with the viruses mainly and nematodes, not with eukaryotic organisms. So this is not a correct statement. ELISA is based on principles of antigen-antibody reactions. ELISA, enzyme-linked immunosorbent assay, which is used to identify the infection of HIV. It is there. The human protein alpha-1 antitrypsin is used to treat emphysema. This is also correct. So the wrong answer here, it is option B. It occurs mostly in the prokaryotic organisms, not in eukaryotic organisms. Next question. Which transgenic microbe is used for cleaning oil spills? Cleaning oil spills. This is called as oil bug. This was first introduced by an Indian-born scientist, Anand Chakravarti. He is the person who identified the whole oil bug. These oil bugs, when they are sprayed in the oil spills, it degrades the hydrocarbons. It degrades the oil hydrocarbons, thereby it protects the aquatic organisms in those areas. It protects aquatic animals. So what is the organism name? This is actually a straight question. The answer is, for this question, it is Pseudomonas putida. Pseudomonas putida. The rhizobium. The rhizobium, we all know that this rhizobium, it is used in nitrogen fixation. And then the Bacillus aminoleucofaciens. This is again a root colonizing biocontrol bacteriums. And this is a source of endonuclease enzyme. A source of endonuclease enzyme. And the fourth option, Pseudomonas fluorescens. The Pseudomonas fluorescens, the bacterium, usually it is not pathogenic, it will not cause any disease. But there are research tells that the Pseudomonas inflorescens, it might cause cancer in patients. It can bring about fever, chills, nausea, vomiting, confusion, and high heartbeat. These are all the functions of the infections caused by Pseudomonas. But the oil bug, it is Pseudomonas putida. Pseudomonas putida. So this is the answer for question number 10. We'll move on to next question. Question number 11 after a break. Children, go for a break for 5 minutes and come back. After the break, I'm going to explain to you about the 11th question. 11th question, as I informed today you are going to write two tests. Two tests. I know it is very difficult for you to stretch yourself and attend the two tests at a time. So at least you can do one test today and the second test tomorrow morning. Or if it is possible, you can complete the both tests by today. So each test carries 90 questions. Each test carry 90 questions in that. So I want you to attend all the questions without choice. Without choice. Okay, children? Because here you can do some mistakes. Try to take some calculated risk. Okay, children? Yes. So now we'll go for a break. After the break, I'm going to explain the rest 10 questions to you. Welcome back, children. Shall we proceed? So I can able to see only Sachin, Shruti and Laya responding. What about others? What happened to Madhavan? What happened to Setu, Darshan? Shall we start, children? Okay, children. The next question is an improved variety of transgenic basmati rice. An improved variety of transgenic basmati rice. There are 4 options are given here. Does not require chemical fertilizers and growth hormones. Any plant, it requires fertilizers and growth hormones are very important. Without that, even if it is transgenic plant, it needs that. And the second option, it gives high yield and rich in vitamin A. Any transgenic food, if it is produced, any transgenic food, or we call it as genetically modified foods, it will be always enriched with vitamins or minerals, or enriched with protein, carbohydrates, like that. Here the transgenic basmati rice, it gives high yield and also rich in vitamin A. The third option is completely resistant to all insect pests and diseases of paddy. When you create a transgenic basmati rice, the transgenicity, it will be specific for a particular disease causing organisms, a particular pathogen. It is not non-specific. It is specific. Gives high yield but has no characteristics aroma. Characteristic aroma, it will be there in basmati rice. So here the option B gives high yield and it is rich in vitamin A, that is transgenic basmati rice. Next question. A gene whose expression helps to identify transformed cell. A gene whose expression helps to identify transformed cell. The first one, the vector. Vectors are vehicles to transfer the genes. And plasmids. Plasmid is actually a vector. The vector. Plasmid, it is a vector which is taken from the E. coli bacteriums. But the question is here, the gene whose expression helps to identify transformed cell. To identify the transformed cell, which is used here in the, if you take a vector, the vector, it has some genes called selectable markers. The selectable markers, it helps us to selecting a right host cell. They are used to identify the selectable markers. It is used to identify the host cell and causes the expression of genes. So the selectable markers are present mainly to identify the correct host and insert the gene and bring the expression of the gene. So the answer for 12th question, it is option D. Option D. The next question, question number 13. Which of the following restriction enzymes produce blunt ends? Blunt ends. Here, there are different types of enzymes are there. Different types of enzymes are there. In the different types of enzymes, you can see it here. These are all different types of enzymes which cut the DNA at different regions. Different regions. But the question is, which one cuts the DNA at blunt ends? If you see it here, SAL1, SAL1, it cuts sticky end. EcoRV, that is the one which cuts the DNA at blunt ends. Whereas HIN3 also sticky end. And XHO1, that is also sticky end. But the blunt cut, like this, the blunt cut, it is done only by EcoRV. EcoRV, the blunt cut. EcoRV is a type of enzyme, endonuclease enzyme, which is taken from E. coli bacteria. It always creates blunt ends. It recognizes the palindromic sequences in the DNA and cut them exactly at blunt ends. Not like this. Blunt ends. So the answer for this EcoR5. EcoR5. The next question. The TAQ polymerase enzyme is obtained from. First of all, we should know what is TAQ polymerase enzyme. TAQ polymerase enzyme, it is used in PCR, polymerase chain reaction. The polymerase chain reaction, there are 3 stages are there. In the 3 stages, they usually differ in temperature. Increase in temperature, sudden falling in temperature. When you are not maintaining optimum temperature, what will happen? Enzymes get denatured. So if you are going to take the organisms, from a normal bacteria, then it won't work out in PCR reactions, the polymerase chain reactions. So we want the extremo bacteria for that, which can live in extreme conditions. This Dermis aquaticus, this is the bacterium which is from which they were extracting the TAQ polymerase. TAQ, Dermis aquaticus polymerase enzyme, is obtained from the bacterium Dermis aquaticus. It is used in PCR reactions. So this Dermis aquaticus organism It is a thermophilic bacterium. This is thermophilic bacterium. Thermophilic bacteria. That's why in polymerase chain reaction they are using this. The next question, question number 15. The two polypeptides of human insulin are linked by. Usually if you see that the human insulin, it has 51 amino acids to be there. There are two chains are there. In insulin, there are two chains are there, alpha and beta. Alpha and beta. The alpha, it has, or we call them as A and B. Chain A, chain B. Chain A has 21 chains, 21 amino acids. Chain B has 30 amino acids. There will be a separate chains. Those chains are linked by a bond. That bond, it is called disulfide bridges. They're called as disulfide bridges. Whereas the hydrogen bonds are formed between the nitrogenous bases. The nitrogenous bases, adenine pairs with thymine. So this bond is the covalent hydrogen bond. Whereas the phosphodiester bond, it is formed between the phosphate and the sugar molecules. In the DNA, the phosphates are linked to the sugar molecules with the help of bond. This bond is called phosphodiester bond. Phosphodiester bond. So disulfide bridges. The next question, consumption of which one of the following foods can prevent the kind of blindness associated with vitamin A deficiency? Here we are seeing four things here: flavor saver tomato, canola, golden rice and BT brinjal. Here this is vitamin A enriched food, and also it protects us from blindness. This flavor saver tomato, this is a tomato which is called genetically modified food. The first food, genetically modified food that is accepted in USA, that is flavor saver tomato. Canola, this is an oil. BT brinjal, brinjal which is obtained from the plant which contains BT gene. This is a transgenic plant which is mainly used, it gives protection to the pest. Pest protected or pest resistant plant. Whereas this one, golden rice. What is a golden rice? The rice plus the carotene gene from carrot, it is inserted here. We know that carrot, it is good for health, which is vitamin A. It can be given as vitamin A deficiency, and also which is good for vision. To prevent the kind of blindness, they are using golden rice. Golden rice means Oryza sativa, that is rice plus beta carotene gene. The beta carotene gene, it will be there. The answer for 16th question is option . Very sorry children. Children, from which question you have not seen? From 14th questions, right? From which question, children? Yes, okay. I'll explain from 14th question again. In the 14th question, the TAQ polymerase enzyme. TAQ polymerase enzyme, we all know that it is used in PCR reaction. PCR. That is to produce the multiple copies of DNA. The multiple copies of DNA, we are using PCR. In that, there are 3 stages are there. In the 3 stages, there will be fluctuation in the temperature. To withstand that, we need an enzyme which is taken from a thermophilic bacterium, the extremophilic. So that's why we are taking from Dermis aquaticus. That's why it is called, T stands for the generic name, AQ, the starting 2 letters of species name, TAQ. TAQ polymerase enzyme. TAQ polymerase enzyme. The next question, it is question number 15. The 2 polypeptide of human insulin is linked by, the human insulin, it contains A chain and B chain. That is, A chain contains 21 amino acids, B chain contains 30 amino acids. So these chains are linked by a bond. That bond they are asking, that bond is called disulfide bond. Because hydrogen bonds are present between the nitrogenous bases. Adenine and guanine pairs with cytosine with triple bond. And phosphodiester bond, it is a bond that is formed between the phosphate and the sugar molecule in the DNA. So the answer for this is disulfide bridges, it will be there. The next question, consumption of which of the following foods can prevent the kind of blindness? Can prevent the kind of blindness. Usually when we are young, our parents will instruct us to take lot of carrot. It is good for eyes, vision. So here, the flavor saver tomato, it is a transgenic food. The first transgenic food that is accepted by the USA government. It is rich in minerals and vitamins. Canola, it is an oil. Mainly, they are used in foreign countries. Fatless oil. And the golden rice. It is a very important source. I'll show the golden rice to you, 1 minute. So you can see the slide. This is the golden rice. See the color of the rice. This rice, it is rice with higher level of beta carotene. Beta carotenes. It will be there. These beta carotenes are good for vision, good for blindness, good for eye vision. That's why The golden rice are used here. The next question. Question number 17. Read the following 4 statements A to D about certain mistakes in 2 of them. You have to find out in 2 of them there is a mistake. I'm going to explain that to you. The first statement. The first transgenic buffalo Rosie produced milk which was human alpha lactalbumin enriched. Here the human alpha lactalbumin enriched milk was produced not from buffalo, it is from the cow, the first transgenic cow. The transgenic cow. And the second thing, okay, the transgenic cow, you can see it here. This is the first transgenic cow, Rosie, in 1997. It produced the milk which is rich in human protein. It contains human gene alpha lactalbumin. This is a cow, not a buffalo. Okay, so now we have identified one there. The second statement, restriction enzymes are used in isolation of DNA from other molecules. Restriction enzymes are used in isolation. What is the function of the restriction endonuclease? Restriction enzymes are molecular scissors. They are called as molecular scissors, which are used to cut DNA, either blunt end or sticky end. To cut the DNA only we are using that. Now we have found the 2 statements are wrong. The third statement, downstream process, one of the steps of rDNA technology. Downstream process, it is a final process in rDNA technology where they'll check for QC, the quality control. The quality control, it will be done. That's called downstream process. Disarm pathogen vectors are used in transfer of recombinant DNA into the host. Usually, to transfer the genes into the host, they'll use certain bacteria which are not harmful. One such bacteria are the Bacillus subtilis and Streptococcus. These 2 are used. So that is also correct statement. So the wrong statements are A and B. So where is A and B? Option B. The next question, question number 18. Genetically engineering has been successfully used for producing genetic engineering has been successfully used for producing transgenic mice for testing safety of polio vaccine before use in humans. Transgenic model for studying new treatment for certain cardiac diseases. Transgenic cows, Rosie, which produces high fat milk for making ghee. Animals like bulls for farm work as they have superpower. So what is the, in that one statement, successfully used for what? The genetic engineering process, first what they have done is, they'll use certain organisms to test the vaccines are to test certain drugs. So the transgenic organisms, the genetic engineered organisms are called transgenic organisms. The first statement, you see the first statement, the transgenic mice are used for testing safety of polio vaccine. Any vaccine, it will be tested with the lower organisms, mice, monkeys, like that. So this is the correct statement. Transgenic model for studying new treatments for cardiac diseases. So transgenic models are nothing to do with cardiac diseases. They are mainly to control genes or genetic related diseases. The third option, transgenic cow Rosie, which produces high fat milk for making ghee. The transgenic cow Rosie, it is mainly made for getting the high protein rich milk. High protein rich milk. Not for making ghee. Here they are used for mainly to make protein rich milk. And animals like bulls for farms to work as their superpowers. Here it is nothing to do with genetic engineering. But this one, the correct answer for this relevant answer is option A. Next question, what is antisense technology? What is antisense technology? Antisense technology. The term anti-sense technology, that means anti means against. Against. So what is this antisense technology? This is RNA polymerase producing DNA. RNA polymerase produces DNA, that is the replication process that is done by the enzymes RNAP, RNA polymerase enzyme, as well as DNAP, DNA polymerase enzyme. A cell displaying a foreign antigen used for synthesis of antigens. A cell displaying foreign antigen used for synthesis of antigens. When there is a foreign antigens, it is used to synthesize the antibodies. Production of somoclonal variants in tissue culture. For production of somoclonal, soma means the clonal, the body, variants in tissue culture, they use embryos, somatic embryos. Different embryos are used. Whereas the antisense technology, it is when a piece of RNA that is complementary in sequence is used to stop the expression of a specific gene. Antisense means against. If there is a gene expression is there, if the gene expression is there, to stop the gene expressions, they are using the same complementary sequence of RNA. To stop the expression of DNA, they are using a piece of RNA that is complementary in sequence. That is called antisense technology. So option A is correct answer for this particular question. Next we will move on to the question, the last question. In TI plasmid, what is TI plasmid? Tumor inducing plasmid. From where we are taking TI plasmids? The TI plasmid, tumor-inducing plasmids, are taken from Acetobacter. Acetobacters are they're used as fertilizers. Rhizobium of the roots of leguminous plant. Rhizobium, these bacteriums are mainly used for nitrogen fixation. Yeast, Saccharomyces cerevisiae. From that, we are not taking plasmids because plasmids are extra chromosomal material. It's an extra chromosomal material present in bacteria. So here, this one, the Agrobacterium tumefaciens. This is the bacterium from which we are taking the TI plasmid, the tumor-inducing plasmid. Tumor-inducing plasmids. Children, with that, we have discussed the questions. Now what I'm going to do is, I'm going to send the papers to you now. Children, you want both the papers now or you want one paper after one paper? Children, you want both the papers now or shall I send one paper, one after the other? Okay, I'll send one paper now. Once you finish that, I'll send the second paper. Okay, children? Yes, I'll send the one paper now. Wait, children, I just attach the paper to tell me whether you got it or not. Children, I want you to, there will be questions there. I want you to start from question number 90. Question number 91, not from 90, question number 91. And one more announcement, children, for you. Tomorrow morning, there is a class. 6:30 to 8 o'clock, there is a class. There is a class. The topic I'm going to discuss is ART, Assisted Reproductive Technology. And then I'm going to do some topics in the microbes and diseases. If you want to attend, you can attend the class tomorrow morning. The class, it will be there tomorrow morning. If you want, you can attend the class. The timing is 6:30 to 8. Anyway, I'll send the link to you. If you want to attend, you can attend. If you want to attend, you can attend. I have sent one paper. You kindly check it. You got it, children? Please check it and tell me. I have sent one paper. Please check, let me know it, whether you got it or not. In that paper, you just start from question number 91. Question number 91. 91 to 180. 91 to 180. Okay, children, all of you got it? Yes. You can start the test. Once you finish the test, send me a message. I'll send a paper tomorrow morning. Because if you do one paper, there is more than enough. I want all the answers today before 9 o'clock. I don't know when you'll start the test. Before 9 o'clock, I want all the answers. Even if you take rest, you can start the test. So before 9 o'clock, I want all the answers. Okay, children. Yes. So thank you very much. We'll see you tomorrow. If you want to attend the class 6:30 to 8 o'clock, you can attend the class in the morning. If not, we'll have the class on Saturday. The next class on Saturday. Next class topic, now itself I'll announce to you. Already I have sent it in the group also. The group also I have sent it. Children, can anyone tell what is the next topic? The next topic is organisms and environment and ecosystem. Organisms and environment and ecosystem. That is the first two chapters in 12th standard. The last fifth unit is there, right? The fifth unit, the first two chapters, it is going to be there. So prepare well. Yes, children? Yes. Thank you. We'll see you in the next class.